All right, so yeah, this is my name, and I work with Bayer. We basically control the LNP vaccine development. So we're in a research group where formulation of LNPs is performed, and we do the analytics. And in this talk, I will focus on the 5-cap and 3-cap analytical methods we use. Unfortunately, similar stuff has, or fortunately for me as well, has already been presented. But yeah, so what are LNPs, or let's say non-viral vectors? So non-viral vectors, I'm sure everyone knows, are used to transport a transgene into a cell, or in our case, a human cell, and both extracellular and intracellular barriers have to be overcome, for which the LNPs have to be optimized. And this is what we do in our group. So the cargo containing an RNA, in this case an mRNA, is transported by a system of lipids, an ionizable lipid to capture the negatively charged mRNA. Then we have helper lipids, which can be also optimized. The composition, cholesterol also influence the transfection efficiency, and also PEG lipids. So what we do the ionizable lipid formulation. As a company, we are used to small molecules, so it's quite convenient for us to do the optimization in-house. We also do the other compound optimization in-house, and for selected organ targeting, for example, we perform ligand decoration. And nucleic acid optimization in terms of maybe codon optimization is also performed. So let's look at the mRNA structure. So it contains, as we had seen before, it's a cap to protect it from degradation, and it also contains a poly-A tail, which is important for the stability. And both influence the translation efficiency. So if we look at the synthesis process of the mRNAs, there are two different synthetic processes. One is a multi-enzyme method, which had also been presented earlier. But in-house, we perform co-transcription with clean caps. And also this reduces the amount of, let's say, non-cap structures which are expected. We get our quality attributes from guidelines. There are many out there. I've removed the USP guideline, which also, you had seen before, contains the capping efficiency and the poly-A tail length. So looking at our repertoire on instrumentation, we have an Andrew Plus. It's not the one outside, but we have one as well. For sample preparation, we want to basically automate as much as possible to reduce human labor and the toll it takes, and also for reproducibility and stuff. We have a few HPLCs. We also have a 2DLC for different methods. And as for detection, we have a BioAccord TOF system. So these instruments are used to control the quality of the drug substance, as well as other lipid components, for which we use LC-CAD, charge aerosol detection. For particle analytics, in-process control, we use dynamic light scattering, Zetasizer, and we also perform RiboGreen for encapsulation efficiency. The drug product itself also has a similar analytics as for the drug substance. So I will start with the 5-cap part. So we implemented two methods. One is the probe-based method from literature, and it's supposed to, so we use a probe to guide the cleavage using RNHAs. But we also have a full digestive method using nucleazepine and also an alkaline phosphatase to remove the phosphates on the nucleotides. We see this improves our chromatography, and the cap denucleotide usually remains intact. And there are different caps which can be used for synthesis. This is the clean cap variations, for example, and they are different in their degree of methylation. So we perform the digestion. According to protocol, we use a probe which is 3 to 27 nucleotides long, and we optimize the separation for 30 mer around according to our expected fragments, which we will create using a DNA mix. The deconvolution works quite well. It's an ion pairing method using our BIACOD system. So the deconvolution is okay. So for the sample preparation, we also had a 4048 oligomer synthesized, which has the same sequence as our mRNA. And for method development, and instead of a cap, we used the uridine. However, we could see that the method works quite well with good deconvolution. We also performed the same method then on an internally synthesized Cas9 mRNA, where we can find the cap1 structure, and for also an mRNA containing 1000 nucleotides, we were also able to see uncapped triphosphate structures. So the method performs its job. However, we wanted to have a non-mRNA dependent method, so basically a universal method. So we decided to go with full digestion, for which we optimized the chromatography. In this case, we can see we used the Waters Premier column first, and then we also used a Korshell column, for which you can see the guanine has very good efficiency, so we decided to go with that, unfortunately for Waters. So we calibrated our signal using external standards. So this method has also the benefit that we can use external standards. We have heard before that certified reference materials for the intact mRNAs are currently not available on the market, so we were also looking for a different method for which we have external standards. And so we did the calibration to estimate linearity, which is quite good using UV as expected. And we analyzed a few of our in-house synthesized mRNAs, which also contain non-standard nucleotides, pseudouridine here, for example, and methoxyuridine here. Here the separation could be optimized, but works very well. And in mRNA-B, which is a non-purified sample, we were able to find also cap structures using UV detection. But of course, the real sample requires higher sensitivity, so we also direct the flow into the mass spec. The mass spec allows us to extract the mass for the clean cap or for the cap1 structure, which is expected. And so the next step would be to see how much of cap we are able to quantify. For this purpose, we did as well a calibration curve. We found that when we use all adducts for calibration, so we have adducts in the spectrum, but if we use all adducts and we also consider the retention time dimension, we can be able to extract the peak volume. And this gives us very good R-square responses. So our limit of quantitation is at below 1 microgram per milliliter, which also means that for higher sensitivity of, let's say, if we need to detect cap structures, which are, let's say, 1% of expected or below 1%, and regulations will be in place probably in the future, this method would require, of course, something like triple quad, so a more sensitive mass spectrometer. So using the UV detection, we are able to do a composition analysis. So we know the sequence of the mRNA. We can then calculate the recovery as in molar ratios, and we can see for the implemented or the contained nucleotides, we have quite good recoveries between 85 and 115%. And our 5-cap method allows us to verify the correct cap structure, because we have also had situations where the, let's say, wrong cap was ordered, mistakes happen, and also the clean caps are quite, let's say, tricky to understand. So the next part will be the poly-A tail as a second quality attribute, which we look at. For this purpose, we digest the mRNA using the RNase T1 enzyme, and to be able to release the poly-A tail. The sample preparation is being developed at the moment, and we use an ion pairing method using BioAccord LCMS system. And for data analysis, we also compared both two different softwares. One is the Waters' own, and one is the Expressionist software, which streamlines data processing, also by gene data. For method development, we used here a poly-A mix. I think it's from Waters, actually, and also it's an ion pairing method. Yes, we do use ion pairing on our mass spec, so it's quite it's contaminated with ion pairs, or we do contaminate it with ion pairs, but we also do cleaning. The deconvolution of the peaks on MS level is quite successful, so we expect good deconvolution for our samples. So we did analyze a technical batch, which is using the MaxEnt and BiSpray deconvolution process. It's maybe important to know that the MaxEnt deconvolution algorithm is supposed to be used above 15K Dalton and the BiSpray below. The poly-A design of this mRNA is a 3070 nucleotide design with a linker, linker nucleotides in between, and we can see that we are able to deconvolute the spectra quite well. But when we have a linker design where the poly-A lengths are similar, then we have a coelution. We see a coelution, and that makes the deconvolution process more difficult, or in this case, unsuccessful. So we decided to look out for a different deconvolution algorithm. In this case, we used a time-resolved deconvolution, and this allows us to deconvolute according to the information on retention time and mass, and this is the poly-A mix which we used. So the process works quite well. If we look at the mRNAs which we use or which we have synthesized, we can see for mRNA A, where we expect 120 poly-As or 120 As tail, we can already see good signals for that range. For the 7030 design, we can also see 2 good peaks. However, in mRNA C, we were not able to detect a poly-A at all, or at very, very low amounts. The digestion protocol involves either magnetic bead purification or not. So we tested both, and we saw that with magnetic bead purification process, we lose information regarding the distribution of the poly-A tail due to probably the washing steps. However, we can see here we have low chemical noise in general. But when we don't use the magnetic bead purification, we can see noise, chemical noise, but the poly-A distribution is detected better. And this chemical noise can be then removed during the data processing. So this is how the results look like. So for the mRNA A, we expect 120 As. We can see at the maximum intensity we have 124 species. But we can also see that we have here a very good distribution, and the distribution length is around 120 nucleotides. For the 7030 design, we would expect after cleavage a GA species and a purely A species. However, we also detect a CA species, and this is something which we can feed back to our mRNA synthesize collaborators. For mRNA-C, as seen before, we don't detect the poly-A, and for SA and SP-Cas9 mRNAs, also 120 A species are expected, and we can be able to detect them. So what we report is the maximum intensity peak for species, and we also report an average value, which is a weighted average according to the intensity of the peak, so that we can be able to kind of determine the polydispersity of the poly-A tail. Yeah. So in conclusion, we use a probe-based method for capping analysis in order to determine the capping efficiency. And depending on the method of synthesis, we can expect cap 0 or not. Structures. However, we also decided to use to implement a platform method for which we can both detect the 5-cap and also use the UV detection for composition analysis. So it's a multi-attribute method, which is easier in handling and more cheaper, because we don't need to purchase any probes. For poly-A, we implemented a streamlined workflow. So we currently use this workflow at the moment. And we found that without magnetic bead purification, we have dirtier samples, but we retain more information about the poly-A tail. So future directions would be to maybe enhance our sensitivity. That means maybe use a different or more sensitive mass spectrometer for detection. And we would also we envision automating these sample preparations using the Andrew pipetting robot. I can see Tobias is already saying yes. And that would be our future directions at this moment. I'm open for questions.